We will have Dr. Savaguru to present for our seminar. Dr. Savaguru received his master's degree from American College in 1989, PhD from Madurai Kamajet University in the area of ecophysiology and abiotic stress in 1995, and awarded a post-fellowship in German from 1995 to 1998, and was a post-fellow in Japan. After that, he received a doctoral degree in Japan, second doctoral degree, and he went to University of Missouri at Columbia as a postdoc from 2000 to 2002, and then he joined IGB in 2002. Yeah, welcome.
Thank you. Thank you for the introduction, Ranji. So, and I thank first the SPIE UAUC chapter President Tony for inviting me to give a talk in this forum. So today I would like to talk about the structured illumination super resolution system rendered by the Zeiss Microscopy microscope system, which is produced by Zeiss. Although there are a couple of companies making this system, in this university right now we have an OMX system in molecular and cellular biology at the CLSL building with the Professor Belmont in charge of that system. And we have a structured illumination super resolution system from Zeiss installed in the IGB. And if anyone of you wanted to come and use that system, you can come and contact me at any time. So today, what I am going to do is I am going to give you a brief overview of what are the resolution limits in conventional optical microscopy techniques, and some imaging tips for this particular sample preparation for the structured illumination system, and the principles of a few super resolution technologies, and we'll look at exactly how Zeiss has implemented the system. I think many of you might have been in the talk with the advanced microscopy course in the last week. Heard talk from Peter Ner from University of Georgia. How many of you were attending that talk? A few of you. So the basics are pretty much what Peter has explained in that talk very well, but I will go through a few of those techniques. But mostly I will concentrate on how the Zeiss system is implementing the super resolution. So as you know, the fundamental limit of light is fundamental limit of resolution is dictated by the numerical aperture. So the lens which collects how much diffracted light is collected by the front lens of an objective determines the resolution, and which is obviously dependent on the wavelength. So whatever the image you are collecting, it depends on the light what you use, and the light dictates how much resolution you get, because we get approximately half the wavelength of light is the typical conventional resolution, which is approximately 200 nanometer, because your visible light starts around 400 nanometer. So half of that is around 200 nanometer in XY, and approximately 700 nanometer you get in the Z. Comparing a couple of different microscopy techniques as of now in terms of super resolution available to people, compared to this is the very commonly used technique in the light microscopy, which is the confocal laser scanning microscope. So that gives you approximately, as you see here in the XY, around 180 to 250 nanometer, and in the Z around 500 to 700 nanometer. But compared to that, you would have heard of a STED, continuous wave STED, or nonlinear STED, or 3D SIM, which is what we are going to talk today. We are going to look into the 3D SIM today. But compared to other techniques, the 3D SIM, particularly when you look at that, it improves the resolution almost twofold compared to the conventional microscope. Better than STED in terms of the Z, compared to palm and stone, which are typical localization techniques. Based on the localization precision, you get up to around 20 to 50 nanometer resolution pretty much with the stone and palm, which is individual labs in this campus are using quite a bit on particularly stone. And there is one STED system is installed in the Loomis. So the main advantage among the compared to any other super resolution techniques compared to the SIM is you can walk with the sample what you use for this conventional wide field fluorescent microscope or a confocal microscope. You don't need to do anything super special to get the resolutions with the structured illumination. That's the major advantage. You don't need to change anything in terms of your labels, your conventional fluorophores or anything. But you need to do some special preparation for both STED as well as palm and stone. But your resolution improvement is only twice in the structural illumination in the XY. So looking at the sample prep briefly, obviously like any other microscopy technique, these are pretty standard. When you look at this, you might laugh at me because there is no optimal sample you can make like what we wanted you to make, because it's a hard in a biological sample. It doesn't behave like how you want it to be. So obviously these are against convention, but basically this is what you would like to have in this microscopy. So you need to have a bright sample, low background, low scattering, and it works best with approximately less than 10 micron in thickness. That's a pretty good substantial number compared to any stone and palm. Those stone and palm techniques works only with 1 micron sample distance, because based on those are pretty much a turf technique. With the STED, in exceptions to STED, and with the STED can go much deeper, but again you don't have improvement in axial resolution with the STED. So there is a caveat on that. So if you use anything thicker than that, then the 10 micron conventional limit, it would be challenging, but there are some ways we can think about how you prepare the sample, how you mount, and stuff like that. We can try to improve the quality. So basically it is rendered in 3 different objectives. The Zeiss makes it usable with 3 high NA optics, but the one what we have right now is the 63x 1.4. And if you need, you can put a 100x, but I would give you a comparison of the different NA and the objectives and the immersion media against the improvement in the resolution in a bit. So typically you wanted to use a highly quality controlled cover slips, 0.17 plus or minus 0.001 millimeter range of correction on your cover slips would be much better, because to avoid the spherical aberration. And anything labeled, any samples labeled at 405 to 642, which you use from DAPI to Sci-Fi, any conventional dyes which you use regularly in your lab can be used in this system. And because we use an oil immersion lens, as of now we have that, so your mounting media, preferably a prolonged gold, because the refractive index would be pretty close to oil when you cure the prolonged gold for over 24 hours. But if you use a buffer, in case if you wanted to grow some live cells and image some live cells, that's also okay, but there will be some spherical aberration. So coming back to this structured elimination, so what is, probably you have seen these basic images several times, but I just wanted to show you one more time. So you have an unknown sample structure. The structured elimination microscopy is fundamentally based on ability to image the high frequency information which is not available normally with the optical system which has a low bandwidth. Okay, how do you transfer the high bandwidth or high frequency information into a low bandwidth optics? What you have? That's what the fundamental principle of this structural elimination. So you have a pattern, a known sample, and then this is an illumination structure you have with a known pattern. When you superimpose these two, before that you didn't see this the pattern what is hidden in this object. So the fundamental principle, what you call this as a Moire fringes. This Moire pattern you see in everyday life in many places. I will show you in few examples. Some fine mesh doors, for example, in this example, as you see, there is a clear mesh here in these two different locations which you don't see with your naked eye. But when you superimpose these two patterns on top of each other in a specific little rotation, you can see actual pattern in this mesh hidden, as you can see when you superimpose it. This is what called Moire fringes. When you actually look at the actual pattern in an individual door, then it matches the Moire fringes. That's fundamentally the same principle used in the structured elimination microscopy. Because this is a high frequency information which is not transferred to your eyes, because we don't get that information because we have a very low bandwidth. But when you use a superimposed structured elimination on structured on top of each other, then you reveal the structure, And this is used in everyday life. If you look at your cell phones in an angle, or if you look at a low resolution photograph zooming in and out of your iPhones, or in your TVs, in a bright light, for example, and if you shine light on your TV, or if you look at the side walls of your home, many places the structure you will see, or a person who is wearing a very fine striped shirt, you can see the Moire fringes on those patterns. So it's used in, say, a high, also in a high security environment, or for particularly to avoid faking, because the Moire fringes plays a very vital role. Because as you can see here in the $100 bill, in many bills, you can see this pattern. And that pattern always matches the actual pattern. When you are in a low resolution, you don't necessarily see the actual pattern, but it gives you an idea that what pattern is hidden. And it is very hard to reproduce that Moire fringes. That's why it's used in bills. So this is another example of how this different intensity of the structural elimination, also it can reveal the high frequency information which is hidden in an original object. So if we go to the frequency space, we have to, because the image acquisition is performed only in the spatial domain. But the calculation for the structural elimination to bring this high frequency information back to the spatial frequency of the actual system is performed using very sophisticated algorithms, fast Fourier transformation algorithms. And the information is calculated in the frequency space. So the circle, what you see here in the frequency space, the diffraction limit of a conventional light microscope, which is 250 nanometer. This is the area of information you normally collect. For an object which is separated at this distance, imagine that's like 250 nanometer, which is resolvable because you are collecting the information to resolve these two objects as two different objects. But when the objects are twice closer than the conventional, then those frequencies are lying away from the conventional limit, what an objective can pick. So these frequencies are not normally transferred into the actual bandwidth what the system collects. So you have a poor resolution. So what you have to do, you have to increase the collection area where you transform or increase the frequency information which is not available in the conventional field has to be transformed from this area into the circle, because your objective sees only this area in a given time. So like around that's approximately like 3 years before my birth, this concept was introduced, and it was revamped by Sun and Lathe in '92. These are the redrawn images of the frequency spectrum from Sun and Lathe. This is the same picture what I showed in the frequency space before in the last slide. So these are the two spots. Imagine those two spots which are unavailable for the optical system. And this is the conventional field of view is imaged by the system, and which is your 1x. And this is the twice the frequency which normally the conventional system doesn't pick up. So the point now is how we will transfer this frequency into the observable field. So what you do using multiple phase images. So you take you need to use a specific type of grading, and using a patterned illumination on the sample. So imagine if you use like 5 different images instead of one image. You collect the 5 different images by moving these frequencies, what you see, which is unavailable here, in a sequential manner. So imagine if I make 5 images in this spectra, and in that 5 images using a 5 different phase grading. And then by shifting the phase grading in a specific direction, I can transform the frequencies, what you see here, in this. Now it is now the area what you see, which is not collected in this area, is moved into this side. And the area which are in this side, which is the frequencies which are on this side, is moved into this direction. So before, what we call is the missing cone in the middle, because you don't collect these two information, is now transferred. By doing that with the different 5 different phases, you can move these frequencies into the observable field. The problem here now is now these frequencies are completely overlapping, because they are not, because the objective is collecting in the same location, but you have the extra frequencies, but they are spatially different locations. So you need to figure out how to put this, the line what you see here and the line what you see here, back in here and back in there. That calculation is done in the Fourier space. So the guy, after 1992 Sun and Lathe paper, nobody have thought about that structural elimination. But after 10 years approximately, Mats Gustafsson from UCSD, unfortunately he's no longer here with us. He just recently passed away. And he come out with this, utilizing this theoretical approach of structural elimination in practicality. So what he did is he, using a fiber, using a polarizer and a specific grading, he split the orders of rays like 0, 1, -1, and +1 orders of rays back into the onto the sample after splitting them using a grading, and then moved using that 5 different phasial images. By moving the grading, he can generate those 5 images to transfer the frequencies back into the back focal plane of the objective. And this lead to, when you look at the conventional frequency space in the missing cone, this is the normal wide field microscope image. This is what typically people call a missing cone, where you don't collect information in the center, which affects your resolution. So in a 2D SIM, if you take the adjacent images, you still have a missing cone information. But instead of 2D, you make this in a 3-dimensional way, And the next slide will show you how the three different locations of those three beams, what he was using, is spatially different in the z direction, as you can see. So then you can register the information back in the back focal plane of the objective, because you can discriminate where these frequencies are coming. Then if you know the location, then you can put back where those frequencies are supposed to be. As you can see here, instead of collecting one sequence of image, you collect 5 phase images. As you can see here, when you superimpose those frequencies in one direction, this is in one particular angle, and you can completely eliminate the missing cone, because the center information is filled right now. And then you do that not only in one direction, you do it in 3 different directions in the sample, so you get an isotropic image compared to an anisotropic image in this case. So putting the information back, how that works is, as you see here, the raw data, and these are the 5 images what I have acquired at 5 different phase gratings. And the raw data looks mixed, as you see here, because every frequency is in one particular location. So then you know, because you use a reference information from the polarizers, you can put back the 0, +1, and +2 light back into the actual frequency. Then you end up in this image. What you see is the 0 order is in the center, which you normally collect with a normal microscope, and the blue and the green frequencies are the added frequencies collected. So in this particular system, in a commercially rendered system, you can do 5 shifts, and the shifts are fixed at 5. But the rotations, you can make this 1, 2, 3 rotations, as you see here. You can make either 3 or 5 rotations. You have a choice. So there is you can extend that 5 rotations in multiple rotations, as well as you can also generate even greater harmonics than, this is a conventional SIM, what we have right now in the picture, what you see here, compared to a conventional microscope. That's the increase, double the resolution in a normal structural illumination linear SIM. But in saturated nonlinear SIM, you can even go beyond the 120 nanometer resolution using saturated structural illumination, where you can increase, this is the conventional field of view, and this is the linear SIM information you collect. And beyond that, the other harmonics can be generated. But you need to use a very high power laser, a pulse laser, in order to generate these harmonics. But I'm not sure how that is going to affect your biological samples. So here, this is a typical rendering of what the microscope sees on the image, and what is happening in the background of that when the calculation is made in the frequency space. So as you can see, this is the typical frequency raw data, where you see the actual frequencies collected, and these are when the grids are patterned illumination. In here, Zeiss uses specific grids, and I'll be showing that in a second, where you can see what are the available angles. When you rotate the grid in a real time, this is how it looks like in the normal live view in the microscope. So basically, what you do once you collected the information and you make the calculation, there are 2 things the structural illumination does. The first thing is in a conventional, this is the theoretical limit of the conventional microscope, 250 nanometer, the circle what you see. The frequency is transferred right now, what you see in your image is not even filling the conventional diffraction limit. That the reason for that is because of scattering and your sample, the refractive index mismatch, and many things contributing that you don't normally collect. If the theoretical limit of a lens is 250 nanometer, nobody gets 250 nanometer in any of their microscope, even in the conventional condition, even if whatever you do to the image to do deconvolution or whatever. So the SIM is filling this conventional limit by, as you can see in the super resolution image, you fill the entire conventional field of view, and beyond that there are frequencies are collected, and that's why these frequencies combined with the frequencies filling the diffraction limit, giving you approximately twice the resolution. So in this system, as you can see, this is the scan head with 405 UV laser and 488, 561, 630, 642 nanometer visible lasers are combined with a beam combiner, and it comes through a grating. In here, we don't rotate the grating, and as a, in the case of Gustafson's original system, but here we use a field rotator. This field rotator, when you move the specific grating, as you know, we do have an option to 0.28 micron to 0.35 micron spacing, grid spacings available in the pattern. This is depending upon, because despite we use only one lens, but the distance in different grid pattern is needed for when you use different wavelength of light, because the resolution decreases with the increasing wavelength, so you have to use a higher grid spacing with the increasing wavelength. So then when you move into the specific grid pattern, the field rotator changes the speed at which the pattern changes on the image, because you cannot wait, you cannot manually move anything in that speed. In order to prevent the photo bleaching, this field rotator helps the changing the angle of the grid. And in a conventional field of view, for example, in a 710 zoom 1 in PMT-based system, that's the field of view you get. But in this system, you get approximately 80 micron by 80 micron with a 63x planopochromat objective. And the field of view is approximately covering a single cell pretty much. And we have a multiple reflector module, all with the bandpass filters, as you can see, for DAPI, FITC, rhodomin, and sci-fi, typical bandpass for all visible channels. The way the images are rendered is there are a couple of ways the Zeiss implements the structural illumination here. So when it depends on your experiment, whether you take multiple Z stacks, or whether you take a multi-channel, or whether you take a time series, for example. So this is like, for example, is a single Z level. So you don't focus anywhere up and down, you just focus to one plane, and then you take a super resolution image in one channel. In that case, you take every rotation 5 images, and 3 rotations in this case here, it will result in 15 images and 30 megabyte of data. And you can also choose to take 5 rotations, then accordingly the size of the images and the number of the images and the size will differ. And this is for a multi-tracking with a multi-channel. Before you saw just a single channel. So in this case, you keep the you finish one particular channel, and then you move on to the next channel in all your rotations. Then when you take a Z stack, again you keep the field rotator in the same position and finish the Z stack. It uses a piezo Z, although on top of a motorized Z, we use a piezo to control to move the sample. Then if you wanted to do a time lapse, we did do some time lapse with a live mitochondria. and it didn't, it didn't disappoint. It was not bad. Actually, as Peter pointed out, the very important thing is in this microscopy to do the live cell imaging is you need to keep in mind what is your depth of focus. Your depth of focus of your object is, if it is within the limit of the 63x planapo 1.4 NA objective, the drift of your objects within, beyond the depth of field will determine how successful your time lapse in super resolution will be. So this is a few examples and tests I have run after the system was installed. And I would like to share that with you with with some examples. So I would like to see whether there is any difference between between the 5 rotations and 3 rotations. And the image what you see here is 5 rotations and 3 rotations. I don't see visually any huge difference. That's a 40 nanometer beads are imaged in there, and as you can see also. Between the line profile of those beads with the different rotations, it doesn't turn out to be a substantial difference in your full width and half maximum. And this is an example you taken with our system here. Between 3 rotations versus wide field, apparent improvement in resolution can be discerned here. This is a cell showing an actin cytoskelet.
Can I ask a question?
Yes?
Have you tried stereoscopic images?
What do you mean with that?
Slightly offset so that you can get a stereo view, you get a 3D effect?
Oh, I haven't made one. Oh, from the obtained images, you wanted to make a 3D kind of a stereoscopic.
You move the objective a little teeny bit, and then you make another image, and you get a 3D view.
Yes, I haven't done that yet. Yeah, I haven't tried. Yeah. But these are what you see here is the 3D projection of all optical sections what I have taken from these images. These cells is a projection of like 12 to 15 slices in the Z. So this is a
monocular image.
Excuse me?
It's a monocular image, though.
Yes, it is a. It's the same field of view, but through the depth. Yeah. This is the previous picture. What you saw is 3 rotations versus wide field, and this is 5 rotations versus wide field. You can see apparently I don't show you the same cell. The reason for that, the sample was bleached because I could not take the same cell. After taking that image, that cell was bleached, so I could not run again the same sample with the 5 rotations. So you can see that when there is a bleaching, you do have an issue. When your sample bleaches, this technique suffers. You keep in mind. Okay, and then this is a side-by-side comparison of the 3 versus 5. And if you look at the apparent histogram, there is no apparent difference between the in terms of the distribution of frequencies. So I think many of you here comfortable and aware of Fourier fast Fourier transformation. Is everyone aware of that technique? So anyway, I just wanted to, I know at least Gabriel I know because I've seen his talk. So okay, so I just for this particularly for biologists, I give this introduction to make a quick understanding of what FFTs does. So we learn particularly, at least my understanding is we learn, FFT is a complex mathematical algorithm which is sophisticated in terms, you can see what, you can get information from some, it is impossible to get from a spatial domain frequency. So particularly we use with Professor Kimani in collaboration. I have learned that from him. And we use that typically for understanding orientation, frequency, and the angle of objects and shape also in an image, which you cannot easily discern with a spatial domain image. Okay, so as you can see here, and these are all nuclei from an experiment where there are different shapes and all kinds of distribution and pattern. So these are the actual FFTs. So when you look at these 4 images, these 4 FFTs, it gives you some information about these 4 images. Number 1, for example, looking at the distribution of the frequency, first thing what you can see is before we go into that, always in FFT, the low frequency information is very close to the center. The high frequency information is away from the center. If you remember that, that will be more help. Okay. So if you look at this nuclei, you can see a horizontal shape and kind of an elliptical shaped nuclei, and it has a pattern and it has a specific orientation. You can see that with the distribution of these frequencies. As you can see, an elliptical shape which determines the elliptical shape of the nuclei. Okay, that it tells that, and it also tells that the frequencies are much lower because it is very close to the center. Okay, but when you have an object like this, for example, is all touching each other, and obviously if you count individual frequencies are much lower, then if you see again the very low frequency, but it doesn't have any specific shape. When you have a random shape, there is no shape to the FFT. Okay, and it is almost when the shape becomes random or the orientation becomes random, the distribution of frequency becomes circular compared to any specific what we call a preferred orientation. Okay, so and when it becomes even more random and the distribution of frequency in this case, because they are not touching each other, then the frequency becomes higher, and still it is circular because it doesn't have any shape. And when you have it higher, then you have a little longer. But still, here you can make out there is a kind of a presence of a shape because some nuclei are some kind of elliptical nature compared to some random also. So that's this kind of information you can discern from FFT. So when you look at an image, the reason I show you that I will be showing many FFT images from the analysis from the structured illumination data, then you can understand what the frequencies are.
Have you been trying to extract things from the phase? Because basically you have a delta function where every object is. So you're just adding a lot of complex exponentials together. Mm-hmm. So if you looked at the phase, you would be more likely to see that.
There is no phase information.
You're only looking at the magnitude.
Yes. Yeah.
You don't have phase information.
Mm-hmm. Yeah.
Why not? I mean, you have...
No, I have not. No, I have not analyzed the phase differences like the way.
Object,
yeah.
So you take the FFT of it. You have the magnitude in the phase. You're only showing us the magnitude. But the phase contains all of the information about where the objects are.
Yes, sir. But in this case, this is only used as just a quick and rough example to show the pattern, not distinguishing the phase and the amplitude. I would definitely think about doing that. Yeah. So this is again the FFT image of the 5 rotations versus 3 rotations now in the frequency space, what you saw before the actual samples. But these are individual planes of those cells transferred into Fourier space. You can see now the isotropic distribution of frequencies in the 5 rotations, where you don't see any modulations on those differences in those angles, but you can see there are some missing information among those circles, and obviously the image is not isotropic. So, but how much that is contributing to the actual image, I haven't done those very detailed analysis, but maybe I need to compare more samples. But as of now, when you look at this more isotropic distribution and an anisotropic distribution of 3 rotations compared to 5 rotations, but the image doesn't transfer, it is not transferred into the actual image because probably I have not used the same image.
Put an image, you made an image such that you populate those areas where the intensity is low, then you will see a difference.
With in these regions, you mean? Yeah. So
you're using an image that doesn't have a lot of energy
in that regions, yeah. Yes. Yeah. So in this system also, it takes approximately, this gives you an idea how much time it takes to, because there are many images you are taking in every plane. You are taking 5 images and at least 15 images if you do 3 rotations to get one super resolution image in one plane. So that is a long time. So people will think how it is, if I have a very dynamic sample, obviously this technique is not for that kind of sample. When you have your dynamics is greater than the speed of acquisition, as you can see, you cannot use this system for highly moving objects. But for statics, even like, for example, the mitochondria, you saw in the videos from Peter, and I do have some videos of mitochondria where if the movement is not that fast, you can reasonably get a decent image of super resolution. So it takes approximately 200, when you do a time series, 250 individual images of 16 super resolution frames at the different Z levels can be obtained with the full frame in 14 seconds. So this is the picture I was talking about the other time, that a comparison of the 3 different objectives and the resolution improvement. So the one what we have is the 63x and the blue squares. So we are getting approximately, using this, 120 nanometer in XY and 300 nanometer in Z. So this is this software which comes with this system. Also, it does, because when you do these modulations, there is a high likelihood of drift or alignment of those phase gratings. If your different wavelengths are not aligned perfectly, then your colocalization, or when you calculate the actual object in a specific given point, will not be perfect. So it comes with a nice multi angle channel alignment. So this is before channel alignment, but after channel alignment you can get a pretty much precise localization of a point object. So this is the same, yes. So this is our 170 nanometer beads, which are used using our system with a conventional wide field, and this is our super resolution imaging combined with the 4 channel colors from blue to deep red. And that's the point spread function of a single bead after the super resolution. So the acquisition of the images in this system is only like 50%. The 50% is calculation. We use very high capacity the computers in order to perform that, because the number of images, the volume of data is substantial when you take like over a time lapse or a Z in multi-channel Z stack. But the program does give you 2 options. Like, one is an automatic structural elimination calculation and a manual structural elimination calculation. So you can do the whole thing in manual in case if you have a complex sample. So there are main functions in this. There are particularly 2 things in the calculation which are done mostly automatically, but you can do that manually too, to compare how the data changes. The particularly 2 things I would like to emphasize here is the noise filter and the baseline cut and baseline shifted. These 2 modality and the super resolution frequency weighting. So these are all with reference to how you shape, like what he was talking, how this modulation in the different phases, how intense they are, whether you want to, how you extract the high frequency and how you suppress the, amplify the high frequency and suppress the low frequency. These filters can play a predominant role, and you might need to pay attention to what you're doing. So this is, for example, a video showing noise filter changes from, it has a range from 0 to -12. So you can look at the frequency space and as well as the spatial domain. And you can see what happens to the frequency when you are going to less negative and when you go to more negative. So it's kind of hard to see in this picture. I have an analysis which I can show you a little better then. And then the frequency weighting, it's also from 0 to 2. As you can see, it's a kind of a sharpening filter. As you can see, how you can see whether you wanted to have an isotropic distribution of the frequency or it emphasizing the frequencies of increased frequencies. If you see too much of emphasis, then you will see more sharpening, or too much of fuzzy if you go either way. So it is normally 1 is the default. So when you look at the actual images to calculate what these functions are doing, so what I did using the same images, I calculated them with the auto calculation, and obviously what you see here, this is the wide field Fourier transformed image. These are the frequencies I'm collecting from the actual system. But when you look at the super resolution calculated image, you can see the enhancement in the high frequency information compared to the conventional wide field. When I use auto calculation with the frequency also auto as -1, you can see almost most isotropic distribution of frequencies with both the lower frequencies and high frequencies. But when I do, what I did is I just go 4 digits lower and 4 digits higher towards positive and negative from the center value. And as you can see in this picture, I lost most of the low frequency information, but I have amplified the high frequency when I go more negative. And when I go to the towards positive, I amplify the low frequency higher than what is actually there, and I suppress the high frequency information. The same thing with the frequency filter. I almost lost most of the obtained super resolution information when I put the frequency filter to -2. But in the case of here, -0.4, it's not a huge difference between the -1 and 0.4. But if you look at the image, you can see the difference. So this is the same information what you saw in one plane. This is for all the planes where you can see this what is going on in much better way, because when you look at the center plane, what is going on in the image in these 3 pictures, it's kind of hard to understand. But when you look at the outer focus plane of the optics, where you can see the fringes of high frequency information, how it is over amplified when you use more negative value on the noise filter, and the low frequency information is suppressed, because way I say that is when you look at the actual, this is the optimal calculation from auto, where you can see the low frequency red spots. Those frequencies are almost nothing in the minor over negative image. But the high amplification of the high frequency is over amplified, and they create noise. And on the other hand, what happened here is all the high frequency information is over suppressed, and with the low frequency information is over amplified. You see more red in the middle, and that amplifies the signal. And how it affects the image, when you look at this image with the same 0.9, and this is with the auto 4.9, you have an optimal emphasis of low and high frequencies. But with the -8.9, as you saw, when the high frequency information is only left and amplified, when there is no low frequency information. This is just the opposite on the other end. And with the frequency filter, this is the optimally processed image, and with the -2, you can see that it becomes more fuzzy, and it is means more smooth and highly over sharpened image. What you get. So between the baseline cut and baseline shift, this is an argument the Zeiss representative told me that some of the reviewers, if you send super resolution images to the publication, the auto mode from Zeiss is calculating this image. With the automatic, it goes to baseline cut. What does that mean is the frequencies up to this point, this is the total frequencies after the super resolution, and imagine the frequency in here is approximately like 7,000 or something, and automatically the system brings the baseline to here and put that as 0 and gives you this image and this information. But when you look at the baseline shift mode, it gives you the entire histogram, and you have the option to change this baseline, because the baseline controls the background. But if somebody wanted to see what is actually in the background of your sample, it would be best to calculate with the baseline shift, which is a manual process. So you can say that this is what you have got. Then that's for the very high, low noise, high signal to noise ratio, samples having very best or good signal to noise ratio images. But for these images where you have inherently high noise, a low signal to noise ratio, what will happen is the image become almost catastrophic when you process with the baseline shift. But the information of this image is still in here. So only it is where the frequencies are cut off. If you bring this bar to here, you get almost the same image from there. And then the cropping function, some people wanted to do the processing very quickly. So when you do that, so you take an image from the total field of view, and then you crop only the sample and do the processing. When you do the uncropped image, because the frequencies, it has an ability to calculate the background from the signal to noise, the gradation is much greater. The accurate calculation of the sample is much better, because as you can see, the objects are much clearly defined. But when you look at the cropped image, the information is pretty much noisy, and it is not as clear as, because you didn't give sufficient information to the system to calculate the background signal to noise ratio. This is same for the beats also there. So then if you look at the wavelength and the resolution, obviously with the 561 nanometer, I use a larger grid space compared to 488 nanometer. Obviously in the same sample, this is autofluorescent pollen, where you can see those the objects. What you see is approximately 250 nanometer in diameter, which is much better resolved with a lower wavelength than longer wavelength. So these are few examples showing desmosomes and keratin connections. As you can see in a regular confocal, you can pretty much make up a few of those, but in very hard places you cannot make up those junctions, which is very clearly resolved with this system. But however, like in any microscopy, you need to keep in mind that the live cell imaging resolution, multidimensionality, versatility, if you wanted to have everything in one, always that's not the, there is no silver bullet in super resolution technique, because every technique has an advantage and a disadvantage. So you will be choosing based on what kind of sample you are using, how vulnerable they are, whether how speed they are moving, how fast the objects are moving, as well as how sensitive to photo damage, because obviously you collect more images than STED, for example, in this system here, whether your sample were able to handle that kind of high intense light for a longer exposure time. That makes a big difference. And with that, I would like to take a few questions. Thank you very much for your attention.
How's the period of the grading chosen?
Based on the wavelength, yeah.
Right. How does it change with objectives? Maybe you can put the slide with the setup back up.
The gratings are to my understanding, the gratings the one which is installed in the system is capable of handling both 100x and 63x.
But if I want to go to 10x, let's say, then I don't need gratings to begin with.
That's right, because the thing is this, the super resolution, because your pixel, even the images, you don't want to. The point is, they wanted to keep you within the, within the resolution. Because the point you come to this system, you wanted to improve the resolution from the conventional resolution of 250 nanometer. So obviously with using a 10x, you are not going anywhere near the conventional resolution. So you will be you might be better off with a confocal, for example, with the 10x, rather than using a super resolution.
I'm just curious, can I double the resolution of a 10x objective? Let's say I only can afford one objective.
Yeah.
Probably those gratings wouldn't work.
I don't think so. Yeah.
But what's the period exactly compared to that?
So it is like the smallest, if I remember on top of my head, 0.28 microns. The smallest grating. So the spacing.
So is it meant to exactly match the resolution?
Excuse me.
Is it meant to match the diffraction spot of your?
Based on the wavelength and the NA, yes. Yes. And it goes up to 0.33 or 0.35 for the 642 nanometer laser. So if you use between, I think between green and orange, pretty much the same. But if you use 405 and 642, there will be a huge difference. So when you use co-localization of two proteins, for example, if you are studying co-localization, I would not use a 405 label and a 642 label. I would use a 488 and a 561. Although they are closer to each other, but my resolution would be, I would not be using a too big of a, my resolution is not apparently, you see in here, between the 488 and actually in here it looks like based on this resolution you would use a 561 and 642 to keep it pretty close to each other. But normally we use these two wavelengths of your probes. Your grating is, but the distance in grid spacing is between here and here is too high than between here and here. So.
So do we have more questions or we can then thank the speaker again?
Thank you.